situated in the anterior funiculus and in the peripheral part of the lateral funiculus. Some of the nerve fibers assume a more or less transverse direction, as for example, those which cross from side to side in the anterior white commissure. But the majority pursue a longitudinal course and are divisible into 1. Those connecting the medulla spinalis with the brain and conveying impulses to or from the latter, and 2. Those which are confined to the medulla spinalis and link together its different segments. That is, intersegmental or association fibers. Nerve fasciculi. The longitudinal fibers are grouped into more or less definite bundles or fasciculi. These are not recognizable from each other in the normal state, and their existence has been determined by the following methods. 1. A. Waller discovered that if a bundle of nerve fibers be cut, the portions of the fibers which are separated from their cells rapidly degenerate and become atrophied, while the cells and the parts of the fibers connected with them undergo little alteration. See footnote. Somewhat later, a change, termed chromatolysis, takes place in the nerve cells and consists of a breaking down and an ultimate disappearance of the Nissl bodies. Further, the body of the cell is swollen, the nucleus displaced towards the periphery, and the part of the axon still attached to the altered cell is diminished in size and somewhat atrophied. Under favorable conditions, the cell is capable of reassuming its normal appearance, and its axon may grow again. End footnote. This is known as Wallerian degeneration. Similarly, if a group of nerve cells be destroyed, the fibers arising from them undergo degeneration. Thus, if the motor cells of the cerebral cortex be destroyed, or if the fibers arising from these cells be severed, a descending degeneration from the seat of injury takes place in the fibers. In the same manner, if a spinal ganglion be destroyed, or the fibers which pass from it into the medulla spinalis be cut, an ascending degeneration will extend along these fibers. 2. Pathological changes, especially in man, have given important information by causing ascending and descending degeneration. 3. By tracing the development of the nervous system, it has been observed that at first the nerve fibers are merely naked axis cylinders, and that they do not all acquire their medullary sheaths at the same time. Hence the fibers can be grouped into different bundles according to the dates at which they receive their medullary sheaths. 4. Various methods of staining nervous tissue are of great value in tracing the course and mode of termination of the axis cylinder processes. Fasciculi in the anterior funiculus. Descending fasciculi. The anterior cerebrospinal, fasciculus cerebrospinalis anterior, direct pyramidal tract, which is usually small but varies inversely in size with the lateral cerebrospinal funiculus. It lies close to the anterior median fissure and is present only in the upper part of the medulla spinalis, gradually diminishing in size as it descends. It ends about the middle of the thoracic region. It consists of descending fibers which arise from cells in the motor area of the cerebral hemisphere of the same side, and which, as they run downward in the medulla spinalis, cross in succession through the anterior white commissure to the opposite side, where they end, either directly or indirectly, by arborizing around the motor cells in the anterior column. A few of its fibers are said to pass to lateral columns of the same side and to the gray matter at the base of the posterior column. They conduct voluntary motor impulses from the precentral gyrus to the motor centers of the cord. The vestibulospinal fasciculus, situated chiefly in the marginal part of the funiculus and mainly derived from the cells of Dieter's nucleus, of the same and opposite side, that is, the chief terminal nucleus of the vestibular nerve. Fibers are also contributed to this fasciculus from scattered cells of the articular formation of the medulla oblongata, the pons, and the midbrain, tegmentum. The other terminal nuclei of the vestibular nerve also contributes fibers. In the brainstem, these fibers form part of the median longitudinal bundle. The fasciculus can be traced to the sacral region. Its terminals and collaterals end either directly or indirectly among the motor cells of the anterior column. This fasciculus is probably concerned with equilibratory reflexes. The tectospinal fasciculus, situated partly in the anterior and partly in the lateral funiculus, is mainly derived from the opposite superior colliculus of the midbrain. The fibers from the superior colliculus cross the median raphe and the fountain decussation of Meinert and descend as the ventral longitudinal bundle in the reticular formation of the brainstem. Its collaterals and terminals end either directly or indirectly among the motor cells of the anterior column of the same side. Since the superior colliculus is an important visual reflex center, the tectospinal fasciculus is probably concerned with visual reflexes. Ascending fasciculi. The ventral spinothalamic fasciculus, situated in the marginal part of the funiculus and intermingled more or less with the vestibulospinal fasciculus, is derived from cells in the posterior column or intermediate gray matter of the opposite side. Their axons cross in the anterior commissure. This is a somewhat doubtful fasciculus, and its fibers are supposed to end in the thalamus and to conduct certain of the touch impulses. The remaining fibers of the anterior funiculus constitute what is termed the anterior proper fasciculus, fasciculus anterior proprius, anterior basis bundle. It consists of a. Longitudinal intersegmental fibers which arise from cells in the gray substance, more especially from those of the medial group of the anterior column, and, after a longer or shorter course, re-enter the gray substance. B. Fibers which cross in the anterior white commissure from the gray substance of the opposite side. End of section 6.